Good morning, everybody. Welcome to the CompBioMed webinar series number 5. I just want to mention that this is the series is run in collaboration with the VPH Institute. My name is Ben Shire. I'm from the University of Amsterdam. Today I would like to introduce our speaker, Adrià Perez. He's a PhD student at the Pompeu Fabra University in Barcelona. He's working in the computational science research group led by Gianni Di Fabritis. Today, Adrià will be talking about high-throughput molecular dynamics for drug discovery. I just want to add a note that just to kind of keep the webinar to run smoothly, to keep your questions for the end. So we'll let Adrià speak, and at the end of the webinar, we'll open up the floor for questions. So if you can just write your questions in the chat box, I will read them aloud to Adrià. So I guess without further ado, I'd like to give the floor to Adrià.
Okay, well, welcome everybody. Thanks for joining us. I'm Adrià Perez, and I'll be presenting this webinar, which will talk about high-throughput molecular dynamics and specially applied to drug discovery. So let's begin. Okay, let's start with an overview of all topics we'll touch in this presentation. We'll first start by introducing drug discovery and the pipeline that it's usually followed to release a new drug into the market. Then we'll start talking about what MD is and what MD simulations are. And then we'll talk about a little bit about computer-aided drug discovery and what's the position of MD between and compared to other computational methods in drug discovery. After that, we'll start going a little bit more deep into MD simulations. We'll review the state of the art. We'll talk about the recent improvements that have decreased the computational cost of MD. Then we'll also talk about Markov state models, which is an analysis technique of simulation data. We'll see what they're about. And we'll also talk about adaptive sampling, which are all these improvements that have helped into coming from single trajectories into high-throughput molecular dynamics. And then after that, we'll go into a practical case where I will show you how to prepare your own simulation to predict the binding affinity between a protein and a ligand. In this case, we'll do it with the example case of benzamidine trypsin, and we'll go over the different steps, which are system preparation, setting up the simulations, and the analysis of the outcoming data. Okay, so we'll first start with drug discovery. So the drug discovery pipeline is the process required to release a new functional drug. And it's mainly based on 2 stages, which the first stage is the drug research and development, the one where you make research in order to find the appropriate molecule to treat a certain disease. And then it comes the testing part, the trials, clinical trials, which has a first preclinical trial using animal subjects. And then we go over the clinical trials on humans to test efficacy and other effects that the drug may have on human beings. And then after that, drug is approved and it's released on the market for its usage. Because we're talking about MD, we will mainly talk about the first part of this pipeline, which is drug research and development, which is where most computational methods are used, and also molecular dynamics, all this special part. And let's continue. So one of the main goals that is important to discover, to test when you're in the research and development part, it's the protein-ligand binding. So is to predict whether a given molecule will bind into a protein target, and if it does, how strongly does it bind. So the potency of the binding effect between a ligand and the protein. There are several ways to test this affinity. Of course, there's experimental assays that can test you that, but we also have computational methods that allows us to predict the affinity between ligands and proteins before going into experiments. And usually these computational techniques focus on extracting these macroscopic properties from microscopic events, and one of those, of course, it's MD simulations. So what are molecular dynamic simulations? So molecular dynamics simulations are a computational tool that allows us to simulate an atomic model of biological systems. In our case would be proteins and ligands, in the case of drug discovery. And we can simulate their dynamic behavior. It's not just that we have the atomic structure, but also their movement, the system movement in atomic scale. It allows us to observe the mechanical movement of our system a long time. That's the important thing about MD. So the strong thing about MD is that it has both a high accuracy, both on high resolution, both on space and time. In this figure here, we can see a comparison between MD, all-atom MD simulations and other experimental techniques that are used either to look for structural information or for kinetic information. And as we can see, the powerful characteristic of MD simulations compared to these other experimental techniques, it's the high spatial and temporal resolution it has, because MD simulations they have atomic resolution, which you cannot achieve with these other techniques. And also that the time resolution is really low. It's in the order of femtoseconds, which you cannot observe in other experimental techniques. It's worth mentioning, though, that MD, although it has a really high temporal resolution, you really need a lot of computational effort in order to extend the other side of the timescale, so to reach higher timescales like milliseconds or seconds. And I want to point out that in the last 10 years, the development in MD simulations and also different analysis techniques and so on has made it possible to be able to detect higher timescales. But we'll talk about this later. So how do molecular dynamics work? Well, it's fairly simple. It's just applying classical molecular mechanics to these atoms and following their movement. So what we have here, it's just the energy functions that describe interactions between atoms. Okay, I won't go into much detail about this, but well, we have different like two different types. There's bonded interactions, not bonded interactions, but whatever. These are the equations that describe the energy between two atoms, and all the parameters of these equations. They're all written in what we call force fields that are used in MD simulations. That usually these parameters are obtained with either experimental techniques or quantum mechanics computations. Okay, but we will use these parameters to compute the energy functions that we'll use to obtain the forces that move these atoms in space. So basically, once you have these parameters, MD becomes fairly simple. It's just applying classical mechanics, which are Newton's equations of motion, to move these atoms along space and along time. So it's fairly simple. So given some initial positions of these atoms, you choose a short time step that you want to move these atoms, which usually is in order of femtoseconds, between 1 to 4 femtoseconds. Okay. And then basically with this time increment and the forces you obtain using the energy functions you have with the specific parameters for different atom interactions, you get these forces where you can obtain the acceleration of these atoms, and based on equations of motion, you just compute the position of atoms after this small time step you have defined. And basically you move these atoms and then you move time forward, and you repeat this process into several steps, thousands of steps, until you have the desired accumulated time, which the longest trajectories that usually go into over milliseconds. Well, we'll go into further detail later. So, well, after this brief description of MD, so what's the place of MD inside drug discovery, and specifically inside the computational tools that are used for drug discovery, which are called which falls under the label of computer-aided drug discovery. Inside the research and development phase of drugs, there's several stages. Okay, first you have to identify the target you want to, well, the protein you want to target with a certain molecule in order to have some effect in the disease you're trying to treat. And well, there's several computational methods that help there in both identifying target and validating the target if there is a pocket where you can target with a molecule or whatsoever. And MD simulations are useful here in order to make conformational analysis of proteins. Okay, but it's also useful in later phases like heat or lead generation, in order to predict binding affinities, as we said. But the thing is, the thing here, usually the method of choice in these stages depends on whether you want a fast method or an accurate method. There's a efficiency accuracy trade-off that usually when you have a fast method, it's usually because it's not accurate enough in certain situations, but in the other case, accurate methods, just as MD simulations, maybe are not suited for high throughput screenings of thousands and hundreds of thousands of molecules. So MD falls into this specific place, which is more of an accurate solution into certain cases, more than a high throughput technique to test, as I said, thousands of molecules. Okay. And here, as an example of what you can do with MD in drug discovery, there's this paper that we released in our lab. This is basically what I'll show you later in the practical case, just with a different, more complicated case. This is basically the binding process of a dopamine D3 receptor antagonist and the receptor, which is a GPCR, which is a common target in pharma research. And the interesting thing in this case here is that because we were using MD simulations, we were able to find a cryptic pocket where this antagonist was binding, that it was not previously known. It was previously unknown where this antagonist bind in the protein, and thanks to MD, we found it. Other techniques that are not accurate, like docking, for example, were not able to find this pocket just because it doesn't allow the flexibility of the crystal structure because it doesn't have any dynamic information. So thanks to MD, because it takes into account the dynamics of the system, and GPCRs are very flexible systems, they were able to open up this pocket that was closed in the crystal structure, and then the binding could happen, and they could obtain all the thermodynamic and kinetic properties of this binding, which is interesting in order to know, for them to find molecules to target this specific pocket that was previously unknown. Okay, so now we'll start. Wait just a second. Okay, we'll start just talking about the recent improvements in MD that has led MD simulations to come from single trajectories. From single trajectories to high throughput experiments, where you use several short simulations that run in parallel, and then they're joined all together. So just to give a little bit of perspective, okay, on how much it has grown. The first simulation of protein dynamics dates from 1977, and it consisted of a 9.2 picosecond trajectory of this small protein, which is BPTI. It's a really small protein. I think it's 20 amino acids, if I'm not mistaken. Really small protein. And it was just a really short trajectory of 9.2 picoseconds. And in comparison, in 2010, a 1 millisecond trajectory of the same protein in explicit solvent, which the previous trajectory was in vacuum. It was only the atoms of the protein there. But this millisecond trajectory is also with usually with a box of water molecules that go around the protein to simulate better the environment that the protein has. And this 1 millisecond trajectory consists of a 1 million increase in trajectory length compared to the first simulation. So this is a huge increase and allows us to study lots of more biological events that can fall into this time scale. And it is in the interest of keep growing that time scale threshold in order for us to be able to study different biological events. So in a recent publication we made in our lab, we predicted based on different publications that we saw that the aggregate time of the total simulation length of different experiments was increasing over time, and we made this sort of prediction that by 2022 we will be able to reach 2020 we could be able to reach the second time scale, which is really relevant for different kinetic for different events like protein binding or protein folding. And one of the main reasons that we're able to reach these timescales is the hardware improvements, software and hardware improvements in MD. So one of them, which is really important, is the implementation of MD codes in GPUs, which has increased the efficiency of simulation, the computational efficiency of simulations by a lot. These are code examples, MD code examples that run in GPUs. Then there's also the appearance, thanks to this implementation in GPUs, the appearance of distributed computing projects like GPU Grid or folding at home that increase a lot the throughput of simulations. And there's also the development of special purpose supercomputers like Anton, which have pushed the threshold of the timescale barrier in MD. And as I said before, all these changes have led we have changed the view in MD of just using a single long trajectory to a more high throughput manner. So high throughput MD simulations is basically spawning different short simulations that then you put all together to analyze. But there's two ways of doing that. You can, like for example, in this naive sampling example where you just start a simulation from a certain conformation, which is imagine this is a binding protein-ligand binding protein-ligand binding example. Okay, and this is the bulk where the ligand is out of the protein. Okay, or for example, a folding example where protein is still unfolded, and you start simulations from there. So you start running some short simulations that will go into different conformations. Okay, and some of them, they reach an intermediate state, which is good because you really want to capture the whole process, so you want to reach the folded state. Then what you can do is just spawn more simulations, this the whole explorer space. And then you can just decide to respawn more simulations, I don't know, randomly or from the ones you started. But this doesn't ensure you that in this second batch of simulations you will reach the same space. Maybe you won't even reach the intermediate space. So it's kind of a non-intelligent way of respawning, of using this high throughput manner of making simulations. So there's this technique called adaptive sampling, which is basically well, it seems it's very intuitive. It's just when you have all your explored space, you analyze your data and try to decide from where you want to respawn the new simulations. So for example, in this example here, one would want to respawn from here, which is the newly discovered space, in order to be able to reach to the bound state, folded state, whatever. So by being able to identify these states, one can just then start new simulations from here, okay? And then it will be fairly easier to discover new states, and which probably are more interesting, and basically making a better usage of your computational resources, so making the MD experiments more efficient. Here's an example on how much this increases the sampling capabilities. Oh, wait. So in here we have an adaptive sampling example, and here is just a single trajectory that keeps running over time. And as we can see here, it just starts 5 different simulations, but from time to time, it just, when these short simulations stop, it stops at analyzing all the data it has. And then it's deciding to respawn simulations from different spaces, probably the ones that have more probability to discover new states. So as we can see, in the same computational time, where the single MD trajectory it's just keep simulating from these minima, here by using a more parallel approach and respawning short simulations, one can explore much faster the conformational space of our system with just this method, which isn't it's an unbiased method, because it's not actually putting in any extra energy into the system or whatever. It's just by making these decisions that we can make our setup much more efficient. Okay, and if we just go further, we'll see that once it has discovered everything, it just keeps analyzing, while the single trajectory, it finally reached this minima. But for example, and we'll see it later with Markov models, we just capture a single transition from here to there, and usually you want to have information from these transitions in order to then have proper kinetic properties computed, to not have to diminish the error of these computations. So let's talk about Markov state models, which are this analysis technique. I'll just go barely over it, explaining a little bit the main idea of Markov state models. So basically, and because of the increase on throughput capabilities of MD, it become more difficult to analyze all this data and to make it humanly understandable and to actually make sense of it. Because on the first trajectories that were made, usually the analysis consisted of actually looking into the simulation with some visualization software. But when you go into scales like microseconds or milliseconds, the human eye, it's not, I mean, just by visualizing them, you don't extract all the information you want. You actually want to know, for example, how much time does it take from one state to go into the other, and that maybe it cannot be done visually. So the coming of Markov state models was really helpful in order to extract these properties, both thermodynamic and kinetic. And we'll see that we will use these in our example of protein binding. Well, basically what this technique consists is you have your set of short simulations, okay, and because this is just several simulations with different frames, and at every frame we have different atomic coordinates. So for every atom we have 3 different coordinates. This is a really high dimensional space, and it's difficult to make calculations there that are computationally efficient. So so what we do here first is to make a projection of these coordinates into a lower dimensional area. So for example, we take the secondary structure at every frame from these conformations or the contact information and so on. And then when we have these data projected into a lower dimensional space, there's also methods to keep further lower the dimensionality, which we'll see in the practical case. And then when we have these data in a low dimension regime, then we cluster them. We can use several cluster algorithms to do that. And then after we have these clusters, which these represent several metastable states of our system, then we compute the transitions between these states to obtain the kinetic properties and therefore also the energetic properties of this. And just to picture a little bit what Markov state models allows us to do, we'll go back to the previous example. Okay, so here all of these came out of a Markov state model, which was able to separate the conformations into 5 different states. Okay, there's the first state, which is bulk, which is when the ligand is further away from the protein, and then we can compute kinetic properties on going into the second state, which is a more intermediate state. It's next to the pocket, descriptive pocket. And then we have this intermediate state, which is 3. And then we can go over to 4, which is the bound state. There's an off pathway. We can also detect off pathways that are not in the main pathway. And that's the power of Markov state models. Okay, and then this is all the theoretical part that I had to explain. Okay, and now we'll just jump into the example case of setting up a simulation for benzamidine trypsin to compute the binding affinity of benzamidine in trypsin. And for that, just after jumping into it, I'll just mention that the main tool I will use to make all of this, it's HTMD, which is a Python programmable environment for biomolecular discovery, and has several different tools to make it easy to set up all these MD experiments. Oh, sorry. It has system building tools like solvating or protein preparing, putting hydrogens. We'll just see now. Also, it has ASMD integrated, which is an MD code to run it on GPUs. But it can be used also with other MD codes like GROMACS or other MD tools. And there's also different deployment systems, like locally on your GPUs, into clusters, or also cloud applications. And there's also protocols for adaptive sampling that we've talked about, but you can also perform standard sampling. It's really flexible. It has also visualization integrated with other softwares you have to download separately, like PMD. And there's also one of the powerful things about HTMD, it's the analysis tools that are put all together, like dimensionality reduction methods like TICA, different projections for our simulation data, like contacts, distance between residues or atoms, surface, accessible surface calculations. There's also Markov state models integrated through this other package called PyEmma to compute affinities and kinetic properties, and so on. And well, just to say that all the material that I'll show, it's also available in the format of tutorials in HTML's webpage. So you can also revisit that in the tutorials on the webpage. So I'll just go out of the presentation. I will go here. I'll be using Jupyter notebooks to run this. And in case somebody doesn't know what are these, it's just a Python interactive environment where you can run Python and interactively, and it's very useful for teaching purposes, like in this case. So we'll just start with our system. Okay, we'll just load HTML and our data. Well, it's the directory. Okay. And we'll start by loading our protein, which will be in this case trypsin. We'll load it into this object, which are called molecule objects. So here we have this molecule object where we can load, for example, PDB files, and it will store all the required information. And we can load it through a local file, or we can just put the PDB code and we will automatically download it from the RCSB servers. So we'll load our protein, and we'll have here a protein view. Because on how I'm doing these notebooks, it won't actually open up anything, but I have this thing ready here. Just wait. This is a spoiler. Okay, so this is our PDB. Okay, this is our PDB file. This is trypsin. Okay, it's the crystal structure of trypsin. This PDB file also comes with benzamidine already bound in it, which is this molecule we can see here. Okay, and we'll now take it off using HTML because we actually want to simulate how it binds there. So if it's already there, we won't get any useful information. So we can see these are the crystallographic waters, and here's the molecule which we'll go now to remove. Okay, we can just the atom names for the benzamidine molecule, it's coming from the PDB file, are called Ben. So we'll just select them using this name resname Ben, and we'll remove the benzamidine. Okay, now it's just removed, and then we'll continue by preparing the protein. So I didn't show it here, but usually PDB files, because it's a crystal structure, they come out without hydrogens. Okay, this is just it just comes with carbons, nitrogens, and just carbons and nitrogens. So it doesn't have hydrogens. We need, in our simulation, to make a proper simulation of our system, we need the hydrogens back there. And as you probably know, protonation of the protein depends on the pH of the environment. So in order to set it up, we have this function here, which is called protein prepare, where we could set up here any pH that we want. For example, I know we can do it in 6. Okay, and it just sorry, no PDB because I didn't load them. Yeah, I load them. Okay, it'll just protonate our protein depending on which pH we've just used. We'll use pH 7 just as a standard. Okay, And then we'll define here, this step is to define segments. So some PDB files come with different chains or different segments that are separated because sometimes the crystal structure doesn't have the enough resolution to define certain loops. So this is useful for when our protein chain is separated into different segments. So, well, this function automatically does it, and this is just to name water and alpha carbons, but it's not necessary. Okay, and we'll center our protein into the origin on the origin coordinates. All right, so this is how, after these steps, our protein will look like. Okay, I'm using this visualization so you can see these white things here. Wait. So now our protein it's fully protonated, okay, and we've removed benzamidine. There's no benzamidine inside it. Okay, so now we have this step covered. Now let's go on with the ligand. Okay, we'll load this PDB file we already have of our molecule. Okay, and we'll also center it into the origin coordinates. And this is how it will look like. Okay, probably you cannot see it. You cannot see benzamidine because it's inside. Okay, now right now this is what we have in our these are our two molecule objects, and benzamidine is in the middle, so it's not useful. And what we want for our simulations is to put this molecule further away, like in any region of the space. So it actually has to bind inside the protein. So that's what we'll do. We'll set the res names and segments. Okay. And first we'll just rotate randomly the molecule so it's not predefined. And then what we'll do is we'll use this function here, which is called max distance, which will compute the maximum distance in our protein. So probably from this point to this point. Okay. This is our max distance in Armstrongs. Okay. And just what we'll do is we'll sum up 10 Armstrongs to this distance, and we'll move our ligand using this function here inside the molecule object. Use this method to move the ligand further away from the protein. So we'll perform this step, and this is what it would look like. Okay, and now we have our molecule in here, and this is where we will start our simulations. You can also generate different positions of your ligand. Okay, and you can start several simulations from different positions of the ligand. That's on your side to choose. And after we've done this, we'll just join all together inside the same molecule object. Okay, in order for the next steps to continue. So we'll just create an empty molecule object, and we'll append the previous protein, the previous molecule object where the protein is, or trypsin is, and also we'll do the same with benzamidine. We'll just do this to prevent equal residue numbers or atom numbers, and they're joined. And well, yeah, basically there's not much to show. We'll just show this. This is our system now inside the same molecule object, is trypsin, and we have benzamidine here, and that's it. And now that we have everything in the same molecule object, we'll continue to solvate our system. So basically put it inside a box of water, because our simulations run in periodic boundary conditions. Okay, we'll just need to make a box around our protein and our ligand, so to make our simulations into explicit solvents, so also simulating the water molecules. We'll just use this function, which is called solvate, and you can use this attribute to set up a box, like a square box, and we'll use the distance we computed before to set up the box, 5 Armstrongs larger than our previous distance. So we'll run this. Takes a little bit, but it's quite fast. And because I already have it loaded here, this is how the solvated system looks like. So we have this box that we have created now with the water molecules, which are these small things. Okay, and what I was talking about, periodic boundary conditions before. So imagine now that benzamidine, instead of going into the protein, goes back here because of the periodic boundary conditions. The molecule, when it comes back here, it will appear in this side, and the same with the other sides of the box. So that's why we just have this reduced environment, and we use periodic boundary conditions. Okay, and after we have solvated our system, we'll just build it using the force field. So basically, we'll calculate the parameters for our system, the required parameters. And well, for proteins, there are already available force fields that have been correctly parameterized. There's in HCMD, they're all. Oh, wait, I messed up. I'll just do it from here. These are all the available files. Okay, you don't need to know much about them. There's also Amber force field available in HTMD, and this is just listing the files. And basically, there's already some defaults defined in HTMD to make it easier. That works with lots of systems. Okay, and because all these force fields are just for proteins. So our ligand, which is benzamidine, it's not defined in these parameters, and you need some external parameters for the simulations to work. There's HTMD provides this tool which is called parameterize, which is used to parameterize these molecules, but that's another topic. And well, and just when we define all the files we need in these lists, okay, we'll just use this function which is called build, just defining the topology and the parameter files. Here you can also, I just don't know what I did, but I messed up my key bindings. But you also can, here you can also set up. You can also set up the salt concentration you want in your simulation and other useful things. So we'll just run this now and it start building our system. Okay. It's putting it in segments because it's what the MD code needs. You see that it's setting the sulfide bonds. And well, it's guessing some other coordinates for missing atoms, and also adds the correct salt concentration. So in our case, it needed only 12 anions in there. So here's what our build system will look like. This is our build system. Well, from here it looks quite the same. But if we take out the waters, we'll see now that it has these different, I don't know what I did. Well, these molecules here, which are the anions required, and here it's benzamidine. Okay, and that's it. After that, we have our system ready to start our simulations. This notebook here. It's to show how to set up simulations. I won't spend much time in here because we cannot actually run simulations now because they take too long. But I'll just go over these a little bit quickly. So in HTMD you also have equilibration protocols, okay, which are usually performed before the actual simulation to just correctly set up the system. Okay, you just import this protocol and you define different attributes like the runtime, time units, which is femtoseconds, temperature of the system, whatever, you don't need to know much about this. The standard protocol works fine to just run a simulation, and then you can look into specific things if you're interested, depending on the case. And then you just, when you have all these things ready, it will set it up in different directories, and then you just run it. You can run it on your local GPU queue. You can also run it in different queues or whatever you're using. And then for the actual simulations, you can just run a production protocol, which will write the necessary files for ACMD to run the simulation. Okay, and it's easy to set up. I will just go. You just set the runtime of your short simulations here, and that's it. And then you can just run the simulations like there are with using the GPU queue, or you can set up, as we talked before, adaptive sampling schemes, which are also provided in HTMD. I won't go into much further technical details in here on how to set up directories and so on. I'll just show that it's fairly easy to set up. You just call this class AdaptiveMD and select different parameters that, if you're interested, you can look about them, projections, and this is for the analysis that it's done in every step of the adaptive. And yeah, and that's it. And you can also set up an adaptive goal with, well, this is, I won't go further into this. And we'll just go straight into the analysis phase. Okay, so here is when our simulations have finished. We have the amount of simulations that we want. Okay, and we'll start analyzing them. Okay, I pre-run some of these commands because they take like 5 or 10 minutes, which is short, but not short enough for this presentation. Okay, we'll start by loading all of our trajectories, okay? Because it was a high-throughput MD experiment. We have several different trajectories here. And we'll load them into this, you usually load them into this using this simlist function that just goes in through all the directories. You can just glop to put all the trajectory directories, and then they'll be loaded into this object here, which then is used by HTMD to perform the calculations to look into the files. So we'll load the files. It doesn't take much long. And when we have our files ready, we'll load it into this metric object when we will start our calculation. So first of all, so our endpoint here is to construct a Markov model of our system. Okay. And so to do that, as I said before, first we need to lower the dimensionality of our space. So we'll start by projecting the atom coordinates into a lower dimensional space. Like, for example, we'll use here contacts between residues. We'll use specifically the contacts between the benzamidine atoms and protein carbon alpha atoms. And that's the metric we will use to describe the different conformations in the different frames we have for the whole set of trajectories. So we'll run this command. It's quite fast. Okay, and it's computing all it's just projecting all these coordinates into this contact metric. Okay, we'll set up the frame step of our simulation, and then we have this plot here, which will plot the trajectory and all the trajectory lengths in our sim list. This is just to detect some trajectories that might be shorter than one we want or what we specified. So they might have some errors, or the simulation might have some problem. So usually we want to take those out just in case they can corrupt our information and our Markov state model. And we can use this function here to automatically drop trajectories that are not long enough as the other ones. Okay, so we have this. Remember that we have this contact projection. Okay, and then we'll use this method, which is which is called TICA. It's time-independent component analysis to reduce the dimensionality of our contact space into lower dimensions that will be the predominant components. It's much similar. It's very similar to a principal component analysis, but the important thing here is that it's well suited to separate the metastable states in our simulations, because it takes into account time, which is what we need here for our simulation data. So we'll just run this. We'll set a lag time of 2 nanoseconds, okay? And we'll project this into all the dimensionality we have with contacts into 3 major dimensions to separate our conformations. So we'll just run this. Oh, well, I started running this, but I screwed it up now. Okay. Well, it will keep running. I think I can stop this because it takes 10 minutes, but we don't need this. Okay, but after this is completed, you can also bootstrap your data. So you can make different Markov models to assess that your results are certain. Okay, and after that, you just proceed to cluster your data. So you cluster your conformations to put conformations together into metastable states, which will be our clusters. And then when you have your space discretized into different states, you just compute your Markov state model using this class here, which is model. You put your cluster data inside here. Okay. Just to remind that you can use several different clustering algorithms. We're using mini-batch k-means coming from sklearn. HTMD provides some clustering methods in it, but you can use, I think you can use any sklearn clustering method for your data. Okay. And yeah, and we'll compute our Markov state model here. First of all, and then before actually constructing the Markov model, we need to choose a lag time where we want to record the transitions. So for example, if our lag time is 1 step, the Markov model computation will look at each frame and the frame after 1 step and look and it would sum up this transition. If we use a lag time of 5, it will look 5 steps further into the frame we're looking at to count the transition. So it is relevant to capture different events or timescales. But ideally, in a Markov state model, this because it's Markovian, it doesn't depend on where you are the total timescales. So to assess the correctness, so the correctness of our Markov state model, we can use this plot, which is called, we plot the timescales over different lag times, and because they should be constant over different lag times we're using for our Markov state models, they should be independent of our lag time. If when we make this plot, if our larger timescales are really flat, like it is in this case, then it means that we can trust our Markov state model. And then we will just select a short lag time, like 5 nanoseconds, when we need to choose a regime where everything is already flat. So it's better to not use really short lag times, but more in this regime. Okay. And also we need to select the macro states we will use. So as we can see, we have this larger timescale in our simulation, and we can differentiate these 4 major timescales from the other really short timescales. And so this might be more relevant to our case than these shorter timescales, which might not be important for us to compute the binding affinity. So we'll just use these 4 major timescales. Well, these 4 slow timescales to be shown in our macro states, which are basically agrupations of microstates, and we'll use 5 macro states to construct our Markov model. Okay, and now we'll visualize them. Don't worry. So I already computed this, so we can just plot free energy surface. This is the free energy surface over the first theta dimensions of our simulation space, the conformational space. We can also plot the states we defined in our clustering, which is this one here. We can see the different macro states, okay? And maybe you're not understanding anything here, but we'll now visualize the states. Usually you could do this to visualize the different macro states, but because it won't work now, I'll just have them, I have them preloaded here. So these are our 5 macro states, okay? This is benzamidine, and we can, this is trypsin, and we can look, and we can see that I have colored, I have colored benzamidine into these different states, okay? So what the Markov state model did, okay, it's to separate all these conformations that we have in our simulations into kinetically relevant macro states, so that we can interpret all this set of data, and basically it has detected these different macro states, and as we can see, they make total sense. So well, let's just stick this here, okay? And we'll go into the kinetics, and then after that we'll look into the visualization. Then you'll see it makes much more sense. So after you have your Markov state model, you can compute kinetic and thermodynamic properties, such as the binding affinity or the kinetics of the binding between benzamidine and trypsin. So we'll just load our model into this kinetics object. You can specify the temperature and the concentration of your protein ligand, whatever, and you just run this. Okay, it auto detects the source and sink state, so from where it starts to where it ends, and we can compute the rates. So which basically are the kinetic properties and the thermodynamic, the binding affinity. So here we have the mean first passage time on, so from source to sink, which is from unbound to bound state. Mean first passage time off, which is going from the bound state to the unbound state, which basically can give us it gives us K on and K off, which these are also useful properties for drug discovery. And then here we have the binding affinity in free energy, which is minus 4.6 kilocalorie mole, which gives us information on how strong does benzamidine bind into trypsin. And we can compare them with different ligands, and we can select the ones that build more strongly into the protein of interest. We can also plot these rates in here, separated on macro states, and we can also plot this into a nice pathway that will be helpful for us. So now that we have this pathway here, I think you can see the. Okay, so we have the pathway here. Okay, our source state it's 2, which it's the least stable state, which probably bulk state. Okay. Yeah, this is macro state 2, which is this one here. Okay, it's the bulk state, which is represented here, but this molecule, this benzamidine molecule, could be anywhere in this space, because this macro state represents the unbound state, the totally unbound state. And then we have different intermediates, and then we have the bound state. So the bound state is macro state 4, which is the one here colored in red, okay, which is the binding pose of benzamidine to trypsin. And then we can see that we have different intermediate states before going into the actual bound state, and that we also have this off-pathway state, which is macro state 0. And as we can see in the pathway, this is the bulk state, and we have these different intermediate states. We can see that this one is less favorable, and this one is more common, but it can also go directly into it. And then we have this off pathway state that it's not inside this this binding process. So it's an off binding pathway. And yeah, and also we can see here by the energies that the bound state, which is macrostate 4, which is this one here, is the most stable one, and then macrostate 3, which is the green one, it's the next most stable one, and then this bulk state, which is this one in the middle here, which can lead us to this is the bulk state, which can lead us into different intermediate states. One is this one here, which then jumps directly into this, but then there's this off pathway state, which is highly energetic, okay, where there's this high energy barrier, and it's not that as much favorable, energetically favorable as this one here. And yeah, this is the power of Markov state models that allows us to separate our simulation data into understandable macro states that we can interpret, and also to obtain these quantifiable properties of our system. And well, that's basically my presentation. This is the end of my presentation, and then if you have any questions, you can put in here. And from my part, that's it. And thank you for your attention.
Yeah, thank you very much, Adrian. Yeah, the floor is now open for questions. If you have a question, you can either post it in the question tab or the chat tab. You can also raise your hand. Okay, I think I don't see any questions. So I just would like to thank the speaker one more time. Thanks, Adria, for the wonderful webinar.
I just want to say that if there's any further question that you may encounter, you can just email me here and ask me any question needed about this. And that's it.
And the slides will be made available on the CompBioMed website, hopefully within the next couple of days or so. So the recording of this this webinar will be there as well. So yeah, I would like to thank everybody for attending and remind everybody that the next CompBioMed webinar will take place on the 5th of December. So I guess with that, I'd like to close the webinar, and we'll be speaking to you on the 5th.
Okay, thank you very much. And see you.